Greetings, Alexandra. Hello. How are you doing?
Hello.
Brains. How are you?
I'm fine, thank you very much this morning, Mark. Yeah, it's a good morning, sun's shining, and we're going to talk about dark things.
I mean, the sun's shining where you are, at least, not here.
Yes.
It never shines here. Anyway, yes, we are going to talk about dark things, and we've been lining this up for a few years now. But life in the universe kept on getting in the way. But here we are, ready to discuss brains and zombies. Now, I should say for the folks at home, Alexandra came to my attention by virtue of her research into the likelihood of brains surviving in the grave, even prehistoric graves, far more frequently, commonly than perhaps we had previously thought. And I remember one of the last sort of calls to action that she gave to us and to you guys as an audience was, be careful with your skulls, check for brains. Do you think that message has permeated the archaeological community somewhat?
I think so. I'm super happy that as we discussed last time we met, Mark, more and more are being found, which is brilliant. We now have the world's largest collection here at Oxford, where I study, more than 540. And I think last time we spoke, we were only at about maybe 200 or 300 odd. So yeah, definitely. And I'm convinced that number is just going to keep growing. So.
Like the zombie hordes. That's it though. So just to be clear, is that actual sort of physical samples, or is it samples and written records?
No, that's actual physical samples. So we have more than 540 brains in our collection, and those are individuals with a whole preserved brain. But actually, in terms of records, there's more than 1,800 reported excavated in the archaeological record since the mid-17th century, which is crazy. And there's more than 400 reported in the fossil record as well, dating back to the Cambrian, some 500 million years ago. Wow. So I feel incredibly lucky. Our collection is about a quarter of the total that have ever been reported. So it's a pretty good data set.
Yeah, absolutely. If you want to look at brains. But today, obviously, it's Halloween week, and we are interested in zombies and the possibility of revenant ghouls rising from the grave to the traditional, brains is what they want to eat. Yeah. But what we presume, and what lots of zombie fiction presumes, is that it's something to do with the brain being reactivated, being fired up in some way, and perambulating a corpse. And I suppose, to be clear, what we're talking about today is very much fiction and speculation, isn't it? It's not remotely trying to imply that anyone could become a zombie.
Absolutely, yeah. But I'm going to take off my scientist hat, and we're going to be imaginative today.
Absolutely. Well, there's some of the best science is imaginative. How do you make that next step?
Yeah.
But I suppose the question is, how have you approached this? What's your first step when I invite you rather sillily to consider zombies?
Well, I did think about this when you first emailed, and on the note of brains, I can't tell you how many email subject lines I have had with that as an opener to my research. But I was thinking about zombies and these shambling, shuffling, flesh-eating, rotting corpses with an insatiable hunger for brains. And I sympathize. As an overworked, underslept PhD student, I am also shambling, shuffling, rotting with an insatiable hunger to understand how they preserve, right? So I was thinking about zombies, and they've changed a lot over the decades, right? I mean, they are a cultural phenomenon in that sense. So, during the Cold War, they were imagined as the product of radiation sickness. And when I was growing up at the height of terrorism in the West, we had 28 Days Later and these insanely fast, rage-filled things that make you think of violence and rioting and civil unrest. But the one thing that unifies zombies through time, right, is the idea that the only way to stop them is to destroy the brain. The Zombieland double tap. And I think that's really interesting in light of the ways in which I imagine that the brain might preserve after death. So the more work I do on these brains, the more convinced I become that while the burial environment might be important in some respects, the brain's unusual and unexpected preservation is something to do with its intrinsic chemistry in life. It's something to do with the brain itself as an organ.
Well, because you were saying when we last spoke that sometimes people buried next to each other, obviously with brains, one person's brain will survive, another brain won't do, and it's even if they're in the same sort of context, chemically speaking. Yeah, very much. So obviously we do think about micro-depositional environments, the things like pH and the availability of metallic ions and pore water content, and all these things can vary at micro scales. But on the whole, it does seem really strange that you could have, for example, a workhouse cemetery or an urban cemetery, something where a lot of people are buried, tending to be quite close in time to each other, but also physically close. And you have a line of graves, one with a brain, a couple without, a couple with brain, a couple without. It's very strange. So I do think it's something to do with the brain as an organ, but that might even extend to the individual level. And we could be looking at individual differences in brain chemistry that dictate whether it preserves or perishes after death, which I think is really cool. So are you sort of therefore sort of touching on a mechanism by which actually a sort of a real world zombie, you'd have some people who are more susceptible to have a brain that would last in that corpse, and other people would just die and perish, and their brains wouldn't continue? I wonder. It's a really good question. So one of the ways that I imagine the brain might be preserving where other organs are decomposing is this idea of molecular misfolding. Okay. So that is proteins and lipids getting tangled up with each other and aggregating in the tissue. And this is one of the things that makes the brain weird amongst organs. It has this unusually high ratio of proteins and lipids. It's 80% water, and the remaining 20% is proteins and lipids in this roughly 1-to-1 ratio. So proteins have a stable three-dimensional structure that underpins their ability to carry out their cellular function. So a protein has a particular job that it has to do, which relies on its shape. But some proteins have regions that lack a stable three-dimensional structure, and these intrinsically disordered regions instead exist as this dynamic ensemble of conformations. And these regions are abundant in the brain because it contains trillions of synaptic junctions and these mind-blowingly elaborate white matter pathways that rewire and reroute as we grow and learn. And those intrinsically disordered regions underpin the brain's ability to do that, which is super cool. So the brain proteome is unusual in this respect, and there are a ton of cells that are long-lived. And in fact, there's even evidence to suggest that these cells might be lifelong. Labeling studies with mice suggest that 90% of their brain oligodendrocytes, which are a type of cell in the brain, could outlive the mice. Wow. So, yeah, that's really strange, right? And we don't see that anywhere else in the body. So what I do suspect that it could be something to do at an individual level, but these things are ubiquitous to the brain in general. I'm happy to chat a bit more about molecular misfolding if you're keen, because it gets quite dark. I mean, I'm more than happy to go there, but it sounds as though initially what you're laying out then is the potential for some brains to have that sort of potential to outlast the body. Again, we're talking very much on fiction and reveling in it somewhat. But I'm just thinking, if this was like a movie scenario, you'd have someone in a white lab coat or something saying, we need to do a test. We need to make sure who's likely to come back, and then you'd have people being like taken off to camps maybe where they'd be monitored, and then obviously there'd be an outbreak, and then so we've got the beginnings of a zombie outbreak, and some people will be susceptible, other people will just be food for the rest, I guess.
It's really interesting that you say that about assessing risk, because actually just this week I've been in touch with some colleagues at a lab in Stanford, and we're going to look at doing some polygenic risk scores from some of our brains from a Victorian site. So it's only 200 years old, but this site is incredibly interesting. It was the burial ground for a workhouse cemetery, but also the nearby insane asylum. So it's quite a dark place. I talked briefly about molecular misfolding, and just to make clear, these mechanisms are akin to that which we see in things like the dementias, but it also underpins a whole host of neurodegeneration. Right. Essentially, the idea is that these protein lipid clumps tangle together, they aggregate, they no longer have a stable three-dimensional structure, they can't carry out their cellular function, so you have this whole host of cognitive dysfunction. The shambling, shuffling things that we imagine about zombies in a way.
So are you suggesting then that the zombie that we're creating wouldn't necessarily be a high functioning brain?
It certainly wouldn't after this process took hold. But as a society, we're terrified of things like the dementias because these molecular misfolding mechanisms are so devastating. So polygenic risk scores are a really good way of assessing someone's risk for things like this to happen. And if brains are preserving in the archaeological and fossil records by a similar mechanism, then it would be really interesting to see if perhaps that starts in life, and being non-enzymatically driven, maybe that process simply continues after death. Wow. And we're seeing, and we've done a lot of proteomic and metabolomic work, so looking at recovering ancient proteins and ancient metabolites from these tissues, and some of the more fascinating ones we've identified from a paleopathological perspective, so a disease perspective, are, for example, alpha-1 antichymotrypsin, which are found in amyloid plaques from Alzheimer's disease brains, myosin heavy chain, which are overexpressed in conditions of severe intellectual disability. We've also found palmitoylethanolamide, this is terribly sad. This is a fatty acid that's highly expressed in conditions of chronic pain and synthesized in response to injury. So it's quite easy to make an imaginative leap there to the way in which we know zombies behave and the kind of horrific trauma, death and burial and reanimation that they've suffered as well. Yeah. It's terribly sad.
It is. And also I'm liking your humane consideration of zombies in that sense.
I worked for years as an undertaker and embalmer before I fell into paleontology, which is what I'm doing now at the University of Oxford. But you can never lose sight of the fact that all of these samples, they were a person, and I find that very frightening as well. I constantly think of, I don't know if you know Baron Samedi, he's the Haitian lord of the cemetery, like the villain in Bond's Live and Let Die. And I'm terrified of becoming that sort of a figure with my collection. So I'm very, very careful to constantly bear in mind that these are people.
Well, actually, to be fair, you raise a very good, of course, a very good point there, but also a point that is at the heart of good archaeological practice. I mean, I all too often I'll receive messages from normally students, fairly green students, where they'll be, I'm digging in wherever, Turkey or the southern United States or something, southwestern US, and there'll be like a photo that they've sent in where maybe they put sunglasses on a skeleton or something. One with sunglasses and a cigarette in their. And they say, share this, it'll be hilarious, that kind of thing. And I always respond in a way which is, I understand why they think this is amusing, but I also try to remind them that these remains aren't, they're not a prop.
Absolutely.
I suppose more to the point, they're not an artifact for them to sort of assess and reconfigure as they see fit. They were a person. And I
hope that these students learn on the job. And having said they're not a prop, I just want to shout out to my amazing colleague Dr. Erin Johnson in pathology, who lent me this desperately distasteful cardigan.
It's excellent. It's absolutely excellent.
Thank you.
Again, we have to be clear, but also we have to allow ourselves the ability to speculate in a way which is strictly, as it were, fictional, as opposed to actually playing with human remains in that sense. But sorry, go ahead.
I was going to say, one thing that I desperately hope remains fiction is I mentioned the proteins that we're recovering from these ancient brains. One of the proteins I'm desperate not to find is prion protein. And I think it'd be really interesting to discuss the ways in which prion disease could play a role in preserving these kinds of brains.
Okay.
So I mentioned briefly this concept of molecular misfolding, proteins and lipids getting tangled up together. The whole concept of misfolding is a biomolecular horror show, but prions are especially terrifying. So they're these aberrant proteins that spontaneously misfold. That's bad enough, given a protein's three-dimensional structure is essential for its ability to carry out its role and activities in a cell and keep the organism functioning. But prions take this to a whole new level of terror. And, if the Midas touch turns all to gold, then prions turn all to darkness.
Right.
They're described as virus-like, and in the context in which we imagine zombies as this contagion, I think, and especially with the idea that the one thing unifying zombies throughout, as a cultural phenomenon that has spanned decades, is that the way to destroy them is to destroy the brain. Prions are a really interesting, it's kind of hypothetical means of creating a zombie, if you like. So prions are described as virus-like because they cause other proteins in their vicinity to misfold as well. And the result is this domino-like cascade of malfunction that spreads like wildfire throughout the brain. They're truly, truly dangerous. And needless to say, prion diseases are invariably fatal. So prions are famously found in the brains of those infected with diseases like transmissible spongiform encephalopathies, things like mad cow disease, which you get from ingesting prion-infected meat. I don't know if you're familiar with there's a disease, Kuru. Kuru was, it spread among the members of a tribe of Papua New Guinea who practiced funerary cannibalism. So deceased family members were cooked and eaten, which was thought to help free the spirit of the deceased. And this awful disease was more common amongst women and children of this tribe because they were the ones that were traditionally consuming the brain, which is the organ in which infectious prions are most concentrated. But actually, you don't have to consume prion-infected meat to get prion disease. It was discovered just last year that they can be aerosolized, so they can be breathed in. Wow. They can fester in the body for up to a decade. So although the chances of working with a brain, ancient or modern, infected with prions is exceptionally low, I'll know maybe by the end of my second postdoc whether I'm doomed. So if I reanimate, make sure you double tap because...
No, that's you actually answered. Of course you did, because you're a very good communicator. But you answered my question that I was about to ask, and that is this notion of whether or not the prions are actually specific to individuals or whether you can pass them on like a virus. But it sounds as though you can ingest them and they'll start to affect cells around them. Do they migrate to the brain specifically, or can they just be anywhere in the body and just affect all protein?
I'm not sure. And there are, and I confess my ignorance here completely, throw my hands up. There are sort of physiologically normal prion proteins, and there are aberrant prion proteins. And my understanding is that normal prion proteins can exist anywhere in the body, but it's not super clear what their physiological function actually is.
Right.
Aberrant proteins, I'm not sure whether they do accumulate in other organs. I haven't found any research to suggest that, but it might be a bias of the work that has been done in diseases like Kuru, because they manifest neurological symptoms extremely early. So it might just be a bias of the fact that we're studying the brain because we're seeing neurological dysfunction. But certainly they do accumulate there. But whether they exist elsewhere, I'm not sure. It's a very good question.
Okay. That leads me on then to what I'm curious then with regards to Kuru. If I'm pronouncing that correctly, is that does that then have a sort of a catch-22 effect where does it almost like viral load? Does it build up in the community because people are consuming ever more heavily, concentrated amounts of this protein?
It does seem to be. If you imagine that the way this virus-like, it's not strictly a virus, but the way that this disease transmits is simply physical vicinity, the more prion proteins you have accumulating in the tissue, the quicker the neurodegeneration will proceed, and unfortunately, invariably, the faster death will result. So it's terribly sad. And
so hypothetically, could this be a mechanism, just going back to our fictional zombie, as to why and how it is that the zombie wants to eat brains? That perhaps it requires either more of the protein, or it requires more uninfected brains to allow that protein to propagate, perhaps?
Absolutely, yeah, that's exactly my feeling as well. It's a very odd thing to hypothesize about, but yeah, maybe that's why they're hungry for brains, just desperate to consume more prion-infected meat.
Interesting. Yeah, interesting stuff.
Yeah. But it does. You would then have a self-limiting population, at least. So there's only so far the contagion can spread, which in a way is a good thing if we're hypothesizing an outbreak of zombies due to prion disease.
Yeah, that is a good thing. But also at the same time, I suppose that also, if I go back to the first premise of there being people who would be susceptible and people who would be, as it were, the food, perhaps the food is a population that if we had, sentient prion brains, that they'd be wanting to pass themselves into, so to propagate into that other population. So, I mean, maybe if the uninfected population kept on having kids, actually it could be endless, potentially. I mean, that's
gosh, you've just made it worse, Mark. That's true. It's awful. Quite possibly. Gross.
Okay, so does it get any worse or any better, or what's the outlook?
I think that's the worst sort of hypothesis I have for how you might create a zombie. Yeah, okay. I think so.
Yeah, but I like
there is one more way in which the brain is different from other organs that I think is interesting in the context of which it preserves. But we're moving away now from the kind of realm of the biomolecular aspect and more into this really interesting field that I recently discovered, and which is really taking off, called metallomics, and the way in which metals interact with biomolecules. So the brain contains neuromelanin, and we're all familiar with melanin, a pigment in skin, most famously. And preservation of melanic pigments is well known from the fossil record. There's fossilized reptile skin and fish eyes, cephalopods, feathers, and there's this whole incredible, field. This stuff dates back to at least the late Jurassic, and it's underpinning this new field of paleocolor and reconstructing, the ancient palette of the dinosaurs and all that sort of cool stuff. But in the brain, neuromelanin is slightly different. So it contains melanic components, which are highly cross-linked naturally. We just spoke about molecular misfolding. It also contains proteins and lipids, which have the propensity to misfold. So you can see how neuromelanin might be a molecule that's susceptible to cross-linking, which is a means of preserving things in the fossil record. It essentially acts to fix tissues in a way analogous to formaldehyde. But neuromelanin famously is known for its ability to collate, to bind and accumulate toxic metals like iron. And iron plays a really interesting role in the brain that I'm very happy to chat more about. Neuromelanin it's neuroprotective, right, in life, and I love the idea that it could be playing a role in preserving the brain after death. So mineralization is a very well-known means of fossilizing a soft tissue. It's the formation of crystalline or amorphous inorganic minerals on an organic substrate, which act to stabilize that substrate. And that explains a great deal of the bias that we find in the archaeological and fossil records toward hard tissues, right? They're already mineralized in life, things like bone and enamel. But it is harder to imagine how an organ, something like the brain, could mineralize. But one of the metallic ions with which organic matter can mineralize is iron. And the brain is really rich in iron, and iron accumulates in the brain during the natural process of aging. The brain needs iron because it's vital for a variety of cellular processes, including myelination of neurons. But it is a potentially toxic metal, so the brain tightly regulates it in life. It's stored in these iron storage proteins, but when iron protein binding is disrupted post-mortem, you get this cascade of redox reactions that essentially lead to the formation of minerals like magnetite. So magnetite is a strongly magnetic mineral that's commonly found in Alzheimer's brain tissues. And we're also finding accumulation of magnetite in our ancient brains, which might explain the difficulty I had in trying to MRI one of these. It was a disaster. It's like I chucked a magnet in an MRI. It was terrible. Right. But yeah, so there's some, it's may not just be the biomolecules of the brain itself, but maybe there's some component of an individual metal load or something that could be playing a role, and the two interacting together are preserving these tissues.
Okay.
It's all to be discovered.
Yeah. Okay. So so again, back to the fictional zombie. They want brains, and maybe they want blood for the iron as well.
Yes.
Possibly.
Mark, you're getting it. You're getting it.
Viscera.
You're making those leaps.
Thank you.
That's it.
Okay, interesting. Well, so so a while ago I was watching a documentary while I was doing some editing, where someone was talking about how The Walking Dead was unlikely because after maximum several months, all the muscles and the sinew and the fibre, everything would just fall apart, and you'd just have bones on the floor, basically. What
if it's mind over matter?
Yeah, exactly. Yeah, just sheer will holding the body together.
Sheer will. Oh. That's why you have to destroy the brain.
That's horrifying. That's absolutely horrifying. So I'm actually really quite thrilled with just how you were given an assignment many years ago now, well, a few years ago, and you've come back with an excellent mechanism by which zombies might occur. I mean, in addition to things like The Walking Dead, I mean, there's also, you touched on 28 Days Later, where there's an emotional, or presumably a sort of a hormone inhibitor that just made people angry. And I think in that sense, that distinction between zombie and infected is an interesting one. But also as well, there's in, for example, computer game fiction, there's things like in The Last of Us, where fungal infection affects brains and makes individuals behave in certain ways. And I know that I'm not asking you to go down this road too far, because this isn't your specialism, but it's interesting how what you find are that there are fungi that focus on specific species. So there'll be a fungus which is directed at a certain type of ant or a certain type of moth or this kind of thing. So that's another sort of freaky real world science possibility. But inevitably, I can't help but wonder what would be your ideal zombie. I sort of touched on this a little bit when I previously interviewed you, but I'm curious, like where would you want it? Where would you want, inverted commas, very strong inverted commas, to see this sort of happen, and how would you want, let's say, in an individual case, the infection to proceed and then the reanimation to occur? So, for example, my ideal would be actually combining this notion of the fast-moving infected with a sort of realistic process of rigor, and then a sort of fluidity as maybe the organs start to come apart and gases build up and so on and so forth, and then eventually something which is a bit more, I suppose, the lucky zombies, if they wanted to continue being zombies, would be something like a Peruvian mummy, where they've sort of naturally just sort of shrunk and their skin is holding them together, but there's still enough sort of electrical activity to get them to shamble. That would be, so initially you'd have a dead person, they'd slowly not ossify, they'd slowly sort of stiffen, so they might be sort of and a bit frustrated as you're running away from them, and then after a few hours they'd loosen up again. What do you think? Do you have any thoughts?
Yeah, actually I do. So okay. I hadn't thought about this at all, because no one has ever asked me what's your ideal zombie, funnily enough. But I love that you just mentioned about fungal infections. And I remember watching Planet Earth when it first came out, and I guess I was fairly young at the time, I suppose, and being just fascinated and horrified by the cordyceps virus. This fungal infection that affects ants, and in fact, there are many of these kinds of awful infections in the natural world. And I find it incredible what these parasites are capable of inducing in their host. bizarre behaviors, almost like mind control. It gets sci-fi super quickly.
Well, there's one that affects snails, sorry, I seem to recall, where the eye stalks become swollen and then they start to ripple with colour, don't they? So it's sort of like...
What?
So they actually affect the tissue to the point that it's sort of, I think it's like red, yellow, green in bands oscillating up the eye stalks just to get a bird to eat it.
That's crazy.
Which, I mean, that...
Wow.
That to me, I mean, maybe I don't know to what extent mollusks like snails could presumably, they're not too distantly related to animals like octopuses and this kind of thing. So maybe there's some latent possibility within the snail's tissue to change colour. But the very fact that the parasite does that, and then it's fascinating. Sorry, as you say, the transformation.
No, that's fine. Yeah. It is. It really is. And I guess like my ideal zombie would be, and I like your point as well about the kind of the biomechanics and thermal kinetics of decomposition. They can't be overcome.
No.
So if we were to imagine a kind of real world zombie scenario, I guess my ideal would be some kind of viral infection that infects the brain and causes the infected to, and I'm thinking in an urban scenario, to climb to a higher height, maybe the top of a building.
Yeah.
By which point they're swollen with decomposition fluids. This virus has replicated in the body. They're stuffed full of this disgusting zombie virus. And they then rain down on the city and infect others. So you have this kind of cordyceps ant-like type where they climb up the stalk and then
and then just
I think that would be extremely effective contagion. And would also tie in appropriately with the means of decomposition that we understand from a forensic science perspective.
Absolutely. But also as well, actually, I think from a sort of a socioeconomic perspective, you're more likely, I think, to find. I'm thinking about like here in Newcastle, but I've seen, when I was last in Oxford, I saw, but also in other large urban environments and towns, the places where there's lots of buildings. So, for example, cranes tend to be those places where people have less of a say in what's being built. So, in other words, it would be the students, it would be the poor, it would be the less, central business district areas that would be susceptible initially to these sorts of scaffolding being used to propagate. So then you'd have like...
We're saying we want this contagion to start in the city, really.
Yeah, exactly.
With the state of the economy, let's start there.
Exactly. Let's start building. Let's start building in the city in London, and our problems will be solved. Okay. So, but what's the end game there? In so much as, presumably from a parasitic perspective, if you have that ant that goes over the colony and rains down on that colony, is it simply to continue finding other colonies is it not self-limiting to a certain extent?
I think it is, and it touches on what we were saying about earlier. I agree with you. I think it probably is self-limiting, but on the whole, that's maybe not a bad thing.
No, not at all. Okay, well,
unless you're deliberately designing a weapon of mass destruction,
which of course,
which I wasn't planning to do this morning.
No, not at all. No. Legal note: do not try this at home. But okay, one final question then. If you were to see that sort of bloated, fluid-laden corpse climbing up a scaffolding or something, how would you deal with it? What would be the best way? Is it like the xenomorph? Like if you touch it, it's going to burst, and there'll be like, if not acid, then certainly infected liquids. How would you stop it from doing its thing?
I was rather thinking, is it the bloaters or the bursters in The Last of Us? I can't remember what they're called. I was rather thinking of those.
Not The Last of Us. It's Left 4 Dead as well.
Oh, Left 4 Dead. That's what I was thinking of. Yeah. That's it.
Yeah.
Well, if I saw that kind of a creature climbing up a building, I think maybe this would be easier in the US, but headshot.
Okay.
Let's just take it out.
Okay. Yeah.
Double tap, That's what I would do.
Okay. And then when it lands and bursts, you just sort of cordon off the area.
Exactly.
And then hose it down.
Minimize the damage. Yeah.
Fair enough. Excellent. I'm so glad that super intelligent people down at Oxford are doing some good work like this. It's good to know. I suppose finally, just on a serious note, because obviously you don't want people to get the wrong impression of your work. What's the sort of some of the best outcomes that you can think of for these sorts of studies? So, for example, I think last time you talked about you briefly mentioned the possibility of being able to identify pre and post literate brains, which was a fascinating possibility. But it also sounds as though you're hinting at the ability to help identify people who might be susceptible to things like dementia long before they get it, or long before they develop it.
I think that it is an intriguing possibility that what we're seeing with these ancient brains may be the extremer end members of aging trajectories that we see in life. So I think aiming to understand the molecular mechanisms underpinning fossilization of nervous tissues, if there is overlap there, then there could well be scope to actually inform some neuroscience, which on some days I think that's insane, and how can we possibly in paleontology contribute to something as valuable as neuroscience, but perhaps that's not as mental as I sometimes think it is. And in paleontology itself, I mentioned that there are more than 400 putative nervous tissues identified in the fossil record, but no one has yet attempted a comprehensive and systematic investigation of how they are still there. And unless we know from a biogeochemical perspective what they consist of now, then we've little hope of attempting to understand anything from a paleobiological perspective. So what they can tell us about things like the evolution of the central nervous system through time and across the animal kingdom, because there doesn't appear to be a bias in the kinds of organisms that we find nervous tissues preserved in. So I think there's huge scope for implications in paleobiology for this work. And more broadly, understanding soft tissue decomposition and preservation might even help us do things like work out where we can locate the earliest animals. These would be multicellular organisms that are entirely soft-bodied. If we can figure out the mechanisms that are taking place to preserve those tissues, the kinds of environments that might be facilitating their preservation, we could target literally globally places where we might hope to find some of the earliest eukaryotes, which is just crazy.
No, it is, yeah. And I suppose and
super exciting. It's what keeps me going day to day with this work.
Absolutely. I mean, globally, but also presumably, off planet as well. You might be able to say, well, this is a candidate location for if you're going to find a multicellular organism. Look there. And I have a ton of colleagues here in Oxford and far more broadly all over the world, much more intelligent than I am and doing incredible things in astrobiology and some of the most ancient, paleobiology as well. And if my work can do anything to facilitate their search for life in the furthest reaches of time or in the furthest reaches of the galaxy, then I'll be happy. Yeah, exactly.
Whether I reanimate or not.
Exactly. I was going to say you can reanimate as a happy person. Yeah, exactly.
That's it.
Thank you so much, Alexandra. I'm so glad we actually got around to doing this. It's
yeah, me too.
Yeah, and also I'm really glad. I'm glad I got. That I made the connections with the brain and the viscera. It's I think you came up with a wonderful proposal there for a zombie.
Might be a chapter in my thesis. You never know.
Yeah. I'll give you a good TripAdvisor score.
Yeah.
If you have any questions or thoughts, folks, please do comment below. I'm sure Alexandra may well check in on the comments. If they're anything particularly pressing, I'll definitely pass them on to her to check out. But have a great Halloween, everyone. Have a wonderful Halloween, Alexandra. And until next time, do take care. Bye-bye.
Bye, everyone.
Oh.